Hello and welcome everyone to part one of a two-part webinar series covering the non-GMO project verification. This is Marcy Burton. I'm a business development manager here for NSF's Consumer Values Verified Business Unit, and shortly I'll be introducing my co-presenter for today's webinar. The topic for today's webinar is non-GMO project Pronto. And really we're going to talk about the basics of the standard and an overview about non-GMO. This webinar series is geared to make your life a little bit easier and get you comfortably and easily on the path to North America's hottest consumer label. So a few points to mention before we get started. This webinar has been started in mute mode to minimize disruptions. If you have questions, use the chat feature in the bottom left side of the screen. Use this chat feature as the presentation goes on, and we will address the questions in the Q&A section at the end of the presentation. And all names will be kept anonymous. And feel free to email either about these presenters following the call. You can see our email addresses are there. So as I said earlier, my name is Marcy Burton, and I'm the Business Development Manager here for NSF Consumer Values Verified Division. I've been with NSF for over 3 years, and I've had some years in-depth technical experience as the technical reviewer for the Non-GMO Project program. And today I'm joined by my co-presenter, Kimberly Tomachik, and she helps develop new certification services here at NSF. And Kim has been with NSF for over 4 years. So you're in good hands today.
Thanks, Marcy, for that introduction. So as Marcy said, this webinar is part one of two, Non-GMO Project Pronto, Standards and Overview. We also offer and recommend part two, which many of you may have already attended. It's called Non-GMO Project Forms and Documents. So part two is really for those companies who have already enrolled with the Non-GMO project and received the application packet from NSF and the accompanying forms. So if you haven't already, you can sign up for additional webinars on our website. Along with the Non-GMO project series, we also cover other certification programs such as gluten-free, which can be bundled with your Non-GMO project verification. So many of you already know NSF because of SQF, BRC, or other food safety certifications. We're a business unit that currently offers non-GMO project verification, gluten-free, and True Source certifications. True Source is for honey. So it's important to note that NSF is not the non-GMO project. We are a third-party technical administrator for the non-GMO project. So that means that we interpret the standard and we apply the rules to ensure that products meet the requirements, so much like an organic certifier or other audit service company. NSF is committed to the professional, efficient, and independent certification activities, and we leave the advocacy work to the advocates. So for those folks in need of a Cliff Notes version of complying with the non-GMO project, this webinar is a high-level overview of the best of Consumer Values Verified non-GMO project portfolio of webinars. So you'll learn about current and emerging regulatory and consumer market drivers, how to analyze your suppliers and supply chain compatibility and compliance, testing requirements and tips, leveraging key non-GMO project variances and other identity preservation quality system components that you likely already have in place. And rest assured, we won't be skipping on the details in this webinar. Think of it more as an accelerated version with an energy drink, just another tool from NSF Consumer Values Verified aimed to get you informed, compliant, and on the path to the hottest consumer label in North America. And we're hoping to do that all within 60 minutes. So you can expect to learn these 7 things today that you see on your screen: more about GMOs, how and why the non-GMO project was created, understand your ingredients, utilize the exceptions, understand your key supply chain. We'll talk a bit about how easy enrollment is, and then we'll talk about retailers and consumers and how they're demanding the butterfly logo. So to get started, let's get on the same page about GMOs. We'll start out with the definition of GMO by 2 U.S. governmental agencies and the Non-GMO Project. So according to the U.S. FDA, Food and Drug Administration, genetic engineering is the name for certain methods that scientists use to introduce new traits or characteristics to an organism. For example, plants may be genetically engineered to produce characteristics to enhance growth or nutritional profile of food crops. And while these techniques are sometimes referred to as genetic modification, FDA actually considers genetic engineering to be the more precise term. Genetic engineering is sometimes referred to as agricultural biotechnology, and the U.S. Department of Agriculture, commonly referred to as USDA, considers agricultural biotechnology as a range of tools, including traditional breeding techniques that alter living organisms or parts of organisms to make or modify products, improve plants or animals, or develop microorganisms for specific agricultural uses. In 2016, the USDA was tasked with the National Bioengineered Food Disclosure Standard and defining what modification means in the context of the new GMO labeling regulations, which are due July 28, 2018. So gene editing and other technologies pose a unique challenge for regulators, as the definitions change and legislative definitions can quickly expire with the next round of technological development. So according to the Non-GMO Project, GMOs, or genetically modified organisms, are organisms in which the genetic material has been changed through biotechnology in a way that does not occur naturally by multiplication and or natural recombination. Cloned animals are also included within this definition. So what is biotechnology? It's the application of in vitro nucleic acid techniques, including recombinant deoxyribonucleic acid, DNA, and the direct injection of nucleic acid into cells or organelles, or fusion of cells beyond the taxonomic family that overcame natural physiological, reproductive, or recombination barriers and that are not techniques used in traditional breeding and selection. So this is just background. You definitely don't need to memorize this in order to get Non-GMO Project verified, but it's good background. Synthetic biology is also included in this definition. So a term you may have heard is SynBio. That's the abbreviation, shorthand for synthetic biology. The development of novel artificial nucleic acid sequences, biological pathways, organisms, or devices, or the redesign of existing natural biological systems.
Great. Now we'll talk about a little bit of the history of the Non-GMO Project and why it came to be. So genetically modified foods have an interesting history, and their development has experienced rapid growth over the last few decades. Within the history, there has been a great deal of controversy and debate about the benefits and risks of GM foods and the production process. And given the current situation around GM foods, the controversy will likely to continue for some time. So we have a few of the key dates in GMO history here on our timeline, and I'll point out a few. In 1973 was when we had the first recombinant DNA organism. It was in 1992 that the FDA declared GE food not inherently dangerous. In 1997, the EU required labeling of GE foods. And in 2005 was the creation of the Non-GMO Project. So you can see a little history of the addition of commercially available crops. In 96 was corn and soybeans, 2011 alfalfa, sugar beets, and industrial corn. And as we know now, there's new coming in like the Arctic apple, potatoes. And right now in 2017, the Non-GMO Project has verified 43,000 products and over 3,000 brands. It's estimated that 64 countries around the world require labeling of genetically modified foods. Countries such as the 28 nations in the European Union, Japan, Australia, Brazil, Russia, and China all have labeling laws. And in 2016, the U.S. joined this list. Through the final definitions and regulations are still in process. We'll talk a little bit about that in this presentation. So the National Bioengineered Food Disclosure Act was passed in 2016. Prior to last year, the U.S. had only voluntary labeling claims. State initiatives that were held in California, Oregon, Washington, and Vermont to require mandatory labeling of GMO foods, were in place. And in July 2016, President Barack Obama signed into law a bill that mandates products disclosure on their labels if there is the presence of GMO ingredients. So this federal law preempts all other state laws, and it's known as the National Bioengineered Food Disclosure Act. So this new GMO labeling law will require foods containing a GMO ingredient to bear a disclosure on the packaging. And this disclosure may be in the form of a text, a symbol, or an electronic link that directly leads to the required disclosure. And there's talks of small food manufacturers having an option for a 1-800 number to provide that additional information or other exemptions. So although the idea seems a bit straightforward, the new law has raised some confusion and many questions, and the USDA is still working on finalizing the definitions that will be held in the Act and in the laws. And the deadline for this new law is July 28, 2018. There was just an open public comment period, and the USDA was seeking clarification on what they would require as genetic modification, whether it would include the new modification techniques or GMOs 2.0, such as gene editing, silencing, and whatnot. And there's also talks whether dietary supplements and animal meat, poultry, and eggs that come from animals that are fed with genetically engineered foods. So much more to come in the next year.
So in light of the lack of transparency outlined in the new law, the Non-GMO Project has committed to continue to work with their participants and the supply chain to provide the highest standard for non-GMO verification and GMO avoidance. The Non-GMO Project is the only independent third-party program that requires ongoing testing of ingredients that are at high risk of being genetically modified. The bottom line is that this new law does not directly impact the Non-GMO Project, nor the right of companies to use the butterfly logo on verified products. The Non-GMO Project started in Berkeley, California, in response to letters from customers who were concerned about a GM soy product. This effort rallied 161 grocery stores and co-ops throughout the United States in a letter-writing campaign to manufacturers of natural food products and supplements in the U.S. The results of the campaign were mixed, with a central problem being the lack of a consistent, industry-wide standard for what non-GMO was defined as. The retailers who started the Non-GMO Project were motivated by a simple idea. They believed that consumers in North America should have access to clearly labeled non-GMO foods and products now and in the future. The basis for the Non-GMO Project product verification program is based in the Non-GMO Project standard. The current standard version 14.2 can be found on the Non-GMO Project website. The Non-GMO Project standard requires practices and processes for controlling GMO contamination by these at-risk inputs and ingredients. Methods such as segregation, traceability, and testing are the core components of the standard. So what do we mean by these three core components? Testing means that all high-risk ingredients at greater than 5% in a finished product formula must be tested in their raw, crop, or seed form prior to use in a verified product, and they must be proven to be compliant with the action threshold. Segregation requirements ensure that once tested, material is protected from contamination and throughout the manufacturing process. And finally, traceability measures ensure that all high-risk inputs are tracked through to the final product. So we'll cover these in more detail later in the webinar.
Great. Next, we're going to look at how the Non-GMO project characterizes ingredients for compliant inputs. So non-risk input is defined by the Non-GMO project standard as materials that are not derived from biological organisms and are not therefore susceptible to genetic modification. This includes materials like salt, lime, and fossil-based products. With non-risk inputs, we must examine the complete disclosure or specification sheet to ensure that the input is indeed a non-risk ingredient and doesn't have high-risk sub-ingredients like an anti-caking agent or whatnot. So a low-risk input is derived from biological organisms, but that are not in the monitored risk or high-risk categories. This typically means the product is a species for which genetically modified versions have not yet been widely commercialized or for which there are no known or suspected instances of contamination. So again, when we talk about single ingredients, it's pretty easy to decipher if a product would qualify as low risk. But it can be a bit more challenging when you are examining multi-ingredient or processed products. So during the verification process, or inputs used in your product seeking verification, documentation is submitted to show the full ingredient composition of inputs used in the products to confirm the risk. So while an ingredient or an input can be low risk, it can only remain that way so long as it is being produced under conditions designed to avoid cross-contamination with GM materials. And one way to demonstrate this is proof that the facility does not use any high-risk inputs. If the facility does use high-risk inputs, you can still demonstrate compliance by showing that procedures and systems are in place that effectively segregate the low-risk input from potential sources of high-risk contamination within the facility. Essentially, we're verifying that the input was produced under conditions designed to avoid cross-contamination with GM materials. So in order to demonstrate compliance, we would need to get these practices and protocols or IP systems such as the BRC certificate, SQF, or other third-party food safety audit certificate. If a supplier does not handle any high-risk inputs, we can accept a statement that they do not handle any high-risk inputs as listed in Appendix B. So certain inputs for which GM organisms are in the research and development stage are considered monitored risk inputs. So these inputs are being developed, but they're not widely commercially available. Or monitored ingredients can also be known organisms where contamination has occurred. And some of these include table beets, Brassica Napa cabbage, apples, potatoes, flax, and mustard. So while these are classified as monitored risk, their requirements for compliance are the same as low-risk inputs. We'll examine the complete input disclosure to confirm the absence of components with GMO risk, including the compound ingredients. We'll verify that the input was produced under conditions designed to avoid cross-contamination with genetically modified materials. Again, one of the third-party food safety audit certificates. Practices or procedures that show that there are systems in place to effectively segregate from potential high-risk sources, or again, a statement that this manufacturer does not handle high-risk inputs, as listed in Appendix B. So now we'll talk a bit about the high-risk inputs. High-risk inputs are inputs for which genetically modified versions are widely commercially available. And this list includes crops, derivatives, and animal-derived inputs. And again, similar to what we discussed for non-risk and low-risk, We talk about single ingredients. It's pretty easy to decipher if it's low risk, non-risk, or high risk. And it may be a bit challenging to examine multi-ingredient and processed products. So the requirement for the standard or for verification, again, is to submit documentation that provides each compound ingredient in the product, and if high-risk ingredients are present, a percent breakdown will be required. So we'll get into that in a bit more detail. So high-risk inputs would be a product or an ingredient which contains derivatives of these high-risk crops or these high-risk crops. They're alfalfa, canola, corn, soy, cotton, papaya, sugar beet, yellow summer squash, and zucchini. And those were the high-risk testable inputs. Then we also talk about the processed inputs or derivatives of those high-risk materials, and compliance must be demonstrated for the high-risk source material. So this is a non-exhaustive list of derivatives with high GMO risk that are commonly used in food production. So it's just meant to provide examples of materials that are considered high risk. Other examples or categories of high-risk ingredients include animal derivatives, bee products, and aquaculture feed. So when submitting products for verification, source materials for these common high-risk ingredients is required. And a few of the other derivatives or high-risk components would be flavors, as they often include carriers or high-risk synthetic biology components, fermentation products such as xanthan gum or yeast products, and vitamins. Vitamins are classified as high-risk ingredients as the source material is often not disclosed, and there's often carriers or standardization ingredients included. So in version 14.2, there's also the non-testable high-risk inputs, and these include inputs that have been modified using gene editing techniques or gene silencing techniques where there is no point in the testing that would target these varieties. And these include canola that's been edited with gene editing, or many of you may have heard of CRISPR or TALENs techniques. Ingredients or substances potentially sourced from synthetic biology. So animal products are also included in the list of high-risk inputs because animal feed commonly contains high-risk inputs. So livestock products and inputs are qualitatively different from any other type of major input verified under this standard, in that there is no point in the production chain at which it's possible to identify GMO contamination using current testing methodologies. So therefore, it is a requirement for the non-GMO project standard compliance to control contamination based on the testing of feed and or seed used to grow the feed. So for animal-derived products at greater than 5% of the finished product, the feed ration for the animals must be submitted. And all major components, major being more than 5% of the feed ration for the animal for the year, is included in the scope. And any component greater than 5% that is high risk must be accompanied with a valid sampling and testing plan. For animal feed, strict testing may be used for the animal feed components, but if there's a positive result, the sample must be sent to an approved and accredited laboratory for PCR testing to ensure that the input is below the action threshold. So in addition to feed, injections of recombinant bovine growth hormone and other hormones may not be used, and cloned progeny. And I also mentioned honey and other bee products being classified as high risk. For bees, the forage area must sufficiently be free of GM commercial agriculture within a 4-mile radius of the hives. This is similar to organic. If the honey is organic, then an affidavit from the organic certifying body that confirms that the honey is compliant to the NOP regulations is sufficient. And if honey is not organic, then maps of the hives showing the 4-mile radius is free of commercial agriculture. GM commercial agriculture is required. So fish and seafood products are also high risk. And when submitting products with these inputs, if they're wild caught, that information must be submitted. And if they're farmed raised, then feed rations are also evaluated. So once an input is defined as high or low risk or non-risk, it's further classified as major, minor, or micro ingredients. So in other words, what percentage is the input in the finished product? If the input is greater than 5%, it's considered a major ingredient. If it's less than 5% but greater than 0.5%, it's classified as a minor. And inputs that are less than 0.5% in the finished product are classified as micro ingredients. Note, all percentages noted are weight percentages of the product, now not counting the weight of salt or added water in the finished product. And the standard also has a defining ingredient rule. If the name of an input is in the name of the product, the input must be treated as a major ingredient, regardless of the amount in the product. Now, as for testing and different exceptions based on these classifications, we'll get into those in just a moment. But with the release of 14.2 of the standard, the Non-GMO project provided some additional information regarding processing aids. So processing aids used in the manufacturing or processing of a finished product or ingredient or input shall be out of the scope of the review if present in the finished product at less than 0.5% and not declared on the retail ingredient panel or the input disclosure documentation of a wholesale product. For the purposes of the standard, fermentation microorganisms are not considered processing aids, and they must be included in the scope. So again, inputs that are less than 0.5% and are not on the retail ingredient panel are out of the scope of review. They do not need to be declared on the formula sheets and not included in the percent breakdowns. They don't need to be compliant, and they will not count towards your micro exemption.
Hi there, sorry about that. I was on mute. I'll go back here. So now we'll talk a little bit about testing. So when is testing required? Testing is required on all high-risk inputs that are in the finished product as a major, that's 5% or more in the finished product formula. So we mainly see this for corn, soy, canola, sugar beets, and animal derivatives. So for example, if you're making a corn chip and the finished product contains 97% corn and 3% oil, we would need to see a sampling and testing plan for the corn. In the case of animal derivatives, let's say a meat-based product that contains 99% beef and 1% spices, We would need to see a sampling and testing plan for the major high-risk components of the feed that the cows ate. There are two types of testing: genetics and immunologically based testing, which is also known as PCR and strip testing. For non-testable high-risk inputs, those are identified in Appendix B, Section B, and Marcy went over those, no point in the production chain exists at which the GM can be identified using current testing methodologies. So an affidavit stating that any such non-testable high-risk input is not the product of genetic modification will be required to establish compliance with the standard. For any non-testable high-risk input, an affidavit must be submitted to NSF for review prior to initial verification and upon renewal as required to ensure compliance with this section. So absence of all GMOs is the target for all non-GMO project standard compliant products. Continuous improvement practices toward achieving this goal must be part of the participants' quality management systems. The key requirement of such quality management systems is to meet and continually be below an action threshold. Inputs that do not comply with the testing requirements may not be intentionally used in verified products. The Non-GMO project has established the following action thresholds for high-risk inputs and products. The established action threshold for high-risk content in human food is 0.9% and animal feed is 5%. Textiles and cleaning products, that's at 1.5%.
Great, so we'll get into a little bit about the different types of testing. So first we'll talk a bit about genetics-based testing. So this type of testing is required before a finished product can be verified. So the frequency and location of real-time or digital PCR testing can be tailored to accommodate your supply chain. The testing must be performed in an accredited and approved laboratory. And there's a list of the approved and accredited laboratories on the Non-GMO project website. Proper controls must be in place. The test must target all commercialized GM events and be from a sample that has been collected from a statistically valid sampling and testing plan. So the sampling and testing plan must be submitted to NSF for review and will include things such as the lot size, the sample size, where in the supply chain the sample was taken, who's responsible for taking the sample, who's responsible for testing, including the lab listed on the Non-GMO project website that you've chosen for the processing. Qualitative PCR may be used. Qualitative PCR basically gives you a positive or a negative. It can be used if the limit of detection is less than 0.01%. and that the lab test confirms there's sufficiently intact DNA for the sample. Now, this must be listed on the sample or the test result that's submitted to NSF. If there is a positive result on the qualitative PCR, quantitative PCR must be used. Quantitative PCR will provide a result as the percent GM content of the sample, and that percentage must be below the action threshold. Each lot of high-risk input used in products seeking verification must be accompanied with lot-specific test results, showing compliance below the action threshold. So in cases where lateral flow strip tests are suitable, and this would be for animal derivatives or animal feed, they must cover all commercialized GM events for the crops in question, and they must have a statistically valid sampling and testing plan, and it should be designed on the basis of risk assessment for the production handling system and shall reflect the level of monitoring appropriate for the risks inherent in the handling system. Analysts must be trained and their performance verified to ensure the tests are reliable. So as part of the sampling and testing plan, you'll put how the sample or the analyst is trained to conduct the testing. For animal feed, the action thresholds are a bit higher. They are at 5%, and for animal food, 0.9%, as Kim mentioned. Now we'll get into a little bit about the key exemptions or special allowances of the non-GMO project.
All right, so first we'll start with the micro exemption. So remember that a micro ingredient is defined as each of which represents less than 0.5% of the product formula and is not a defining ingredient, so it's not in the product name. So the micro ingredients, the scope of review for these ingredients includes application of the limits in Section B below, which shall be limited to the input used directly in the product as opposed to growth medium or feed. So the following inputs are not allowed if they are the direct product of genetic modification. For finished retail goods, if they're listed on the ingredient panel, or for products sold without retail labeling, if they're listed on the input disclosure documentation. So any added nutrient, vitamin, or other active component contained in a finished product supplement product must be non-GMO, and that restriction actually doesn't take effect until May 2019. Then we have viable microbes, functional enzymes, and products of synthetic biology. So all of those cannot be used in verified products through this exemption. So any given product formulation included in the program must not contain more than 10 exempt microingredients or 0.9% total exempt microingredients. Formulations exceeding 10 microingredients or 0.9% total must either be reformulated or enough of the micro-inputs must be verified as non-GMO project standard compliant in line with the standard in order to reduce the number of exempt micro-inputs to 10 or to 0.9% total or less. So the numerical limit of 10 on exempt microingredients expires on May 20th, 2019. In cases where GMO analytical certificates or traceability linked to analytical certificates of the raw precursor material is not available, non-GMO project compliance status of minor and microingredients can be verified based on affidavits from suppliers as long as these ingredients are the product of a system that has been designed to avoid GMOs. So those might be your identity preservation systems. Examples of these systems are organic certification and other non-GMO identity preservation systems. Suitability of these or other identity preservation systems are subject to review by the technical administrator, that's NSF in this case. Additional information might be required to demonstrate how the system ensures GMOs are not used and that there's sufficient segregation and traceability in place. Suppliers must agree to provide further information or demonstration in support of the affidavit when requested. And when available, valid certificates from third-party certifiers are acceptable alternatives to affidavits under Section 6 of the standard when third-party certification programs satisfy the requirements for which an affidavit would be used. So again, that's an example of that is the organic certificate and summary or another non-GMO IP system. So now we'll talk about downgrades. An example of something that could be downgraded would be cornstarch produced in a country where GMOs are prohibited from growth or import. The non-GMO project standard compliant seed was confirmed as having been used, and documented identity preservation procedures are in place for the manufacture and transport of the input. So these are on a case-by-case basis. Certain high-risk inputs can be downgraded to low-risk status based on source, documentation, protocols for contamination prevention and avoidance, and or laboratory results demonstrating consistently low risk of GMO contamination in accordance with the standard. And individual inputs may only be downgraded by the technical administrator, that's NSF, with the approval of the non-GMO project.
Great, Kim. So key to being successful on your non-GMO project journey is really understanding your supply chain and their ingredients as well as the operations in place for which their ingredients come. So as with the ingredients, you can expect to submit information about your operation and your practices of segregation and traceability. And now we'll get into talking a little bit about facility inspection. So unless the producing facility is exempted from inspection, all facilities are required to be inspected. So you may not have to undergo an on-site inspection if you are producing products in which there are only low-risk inputs, or if your products are using low or high-risk inputs that are either exempt from evaluation or approved via the supplier affidavits. So if your products are being produced in a facility where there's parallel processing of major high-risk inputs, that is when an inspection is required. So parallel processing is if you have a finished product that has a high-risk ingredient at more than 5%, and in your manufacturing facility you have a compliant and non-compliant version of that input. That's defined as parallel processing, and that will trigger an inspection. So again, if your facility is dedicated to non-GMO project production and all inputs are found to be compliant, an inspection is not required. If it's only low-risk ingredients used in your product, an inspection is not required. If you're using a contract manufacturer for the first 3 years of verification, an inspection is not required as long as you have a plan to bring the contract processor into participation within 3 years or sponsor an on-site inspection after 3 years. And products of a facility that are dedicated to organic production, if no parallel processing of high-risk ingredients is occurring. So I wanted to talk a little bit about the contract manufacturer or co-packer exemption. So in the standard, any manufactured product that is made by an operation contracted by the participant may be evaluated and approved under the program as long as it is a product of a system that has been designed to avoid GMOs. So some of those systems, again, are the organic certification or other identity preservation systems. The contract processor's exemption from inspection expires after 3 years unless otherwise exempt from inspection. And we see contract manufacturers become participants, and in that case, if they have parallel processing, inspection will be required, or often we have brand owners apply as the participants. So what should you expect if an on-site inspection is required? So the operation will be assessed to confirm consistency with the documentation that you submitted to us for review on how you intend to comply with the non-GMO project. So we're inspecting to confirm that the policies and procedures you documented on paper during the application and desk audit phases are truly what you're doing in practice. So the inspector will verify the efficacy of the compliance plan, the segregation and trace back systems, and that all necessary record-keeping requirements are in place. The inspector may comment on critical control points and how commingling and contamination may be prevented at these points. However, it's important to note that a non-GMO project inspection is not an audit. The inspector may not consult or make a compliance decision. The inspector will return their findings to NSF, and it's NSF's responsibility to issue any corrective action and to determine compliance. The components of the compliance plan that will be requested in the application review, Kim touched on them at the beginning of our presentation, are traceability, cleanout, and segregation. So each lot of non-GMO project verified product or input must be traceable back to specific lots of the inputs used in its production. So during our review, we'll look at the system you have in place to track inputs from receipt through processing and out to final sale. So we know this varies by facility, so you'll submit what you have in place. Traceability records should explicitly trace and track the non-GMO project standard compliance status of both inputs and final products. And you should anticipate for this documentation to be requested during the audit and during your review. This can include mock recalls or traceback exercises. So the aim of cleanout and segregation procedures is to prevent GMO contamination of inputs, works in progress, and final products. The receiving, production, processing, and manufacturing, transfer, and storage facilities, as well as shipping and transportation conveyances, should be inspected, cleaned, purged as needed to remove sources of GMO contamination, and all relevant cleaning, purging, and inspections shall be documented. So if this operation is dedicated strictly to non-GMO project standard compliant production, then segregation measures within the production operation are unnecessary, and segregation measures are required if the facility is not dedicated to non-GMO project verification, and they are critical where any products are manufactured on shared lines.
All right, so if you're not already enrolled in the Non-GMO Project Verification Program, you might be asking yourself how you can go about getting enrolled. Well, fortunately, enrollment is very easy. If you're interested in getting your products verified or adding additional facilities, you can contact us at NSF for an initial application and a customized cost estimate. And now I'll walk you through the 6 steps to the verification process. So step 1 is application, document submission, and quoting. In the initial stage of the process, we'll collect basic information about your products, ingredients, and facilities, and this will give us a better understanding of your business so that we can quote you properly and provide you with specific documentation requests to assess your ability to comply with the non-GMO project standard. And during this step, you'd likely be working with Marcy as our business development manager. Marcy will provide you with a quote that's customized to your operation. So then we have step 2, that's pre-assessment review and report. At this point, an NSF evaluator will review your submitted information. In the pre-assessment report, you may be asked to provide clarification or more documentation. Then step 3 is the on-site inspection if necessary. If applicable, you can schedule an on-site inspection with your certification project manager. The inspection can be combined with your other inspections through NSF, such as organic certification or gluten-free, SQF, BRC, or Canada Gap audit. Step 4 is the desk audit review. An NSF evaluator will review your replies to the pre-assessment report and any findings from the inspection if you had an inspection. If any non-compliances are found, NSF will alert you and give you time to resolve them. So then we have step 5, which is the non-compliance resolution phase. So if there were any non-compliances found, an NSF evaluator will have described what is needed to ensure full compliance, and your certification project manager can help you resolve any remaining non-compliances. So once those are corrected, a verification decision can be made, which leads us to step 6. So once you've successfully completed the verification process, you're verified to the non-GMO project standard, and you have the option to incorporate the non-GMO project verified seal on your packaging in accordance with your licensing agreement that you'll keep with the non-GMO project.
Great, so now we've earned the logo. The non-GMO project claim and butterfly logo is a solid option for a verified claim for GMO avoidance in North America. And it's the fastest growing label in the natural channel by a wide margin. So in terms of the growth of the non-GMO project label, there are currently more than 3,100 brands making more than 43,623 products that have chosen to achieve this claim. And what's their reward? Well, sales of non-GMO project verified items have been on an extreme growth curve and approaching $24.5 billion annually. And it's important to note that the current non-GMO project licensing agreement is for products to be marketed in the U.S. or Canada. So use of the Non-GMO Project logo is not allowed in packages in other countries. And if that is happening, there is an international addendum that must be signed with the Non-GMO Project. In April 2017, Greener Choices, the Food Safety and Sustainability Center at Consumer Reports, released a comprehensive assessment of the Non-GMO Project verified sale, categorizing it as a highly meaningful label for consumers wishing to avoid GMOs in the food they buy and support farmers who don't use GMOs. So this slide just shows that about 1/4 or 26% of consumers across all grocery shopping channels sought products making non-GMO claims. And this was a report from 2015 for a Grocery Shoppers Trend Report. And we can see that that's growing. The projected growth of the non-GMO labeled products is expected to continue at a healthy 5 to 10% rate into 2020. And that's according to estimates by the Nutritional Business Journal. And it is confirmed that Whole Foods is holding true to their deadline for September 2018 to have the non-GMO claims on packaging. And finally, there is considerable amounts of consumer research indicating that awareness of and concern about GMOs is high across the globe. The consumers are increasingly concerned with how GMOs impact their personal and environmental health. So now we have completed the webinar, and we are happy to answer questions that you may have. So you may use the chat feature in the